Okay, well, welcome back. Well, I actually recognized also in the tutorials that we had that quite few of you are also working or planning to work with bacteria and AFM. And so I would like to show you some of the methods that we are using. So, I mean, bacteria are certainly also interesting from an AFM side because they're structurally interesting. Now we're here, thank you. Okay, because they have a dedicated structure. Some of them, particularly the gram-negative, have 3 layers, and the proteins in each layer have unique functions, and they are essential for cell interactions, for instance, with the environment. And bacteria can also be mobile. We were also discussing this yesterday, and of course the challenge is then to how can we tackle the mobility with fixation and so on. And they have structures that they are using for their motion, like flagella, for instance. And so they kind of vary a lot in their composition and function. Then together with bacteria, what is also important is biofilms, which can also be followed with AFM, so adhesion, microcolonies, and then maturation. And so bacteria also have the ability to adhere to surfaces to form biofilms. And this community serves then as a protection against the environment. So the question is how they adhere to the surfaces. It depends on how the nutrients are available, how hydrophobic, how hydrophilic they are, and then also on the electrochemical properties of the bacteria, so how they can attach. And they first for microcolonies and then macrocolonies before the biofilm can be finished. And there is, well, I already mentioned flagella, but some other bacteria, for instance, also carry pili or fimbriae, and there's also non-pilous adhesions. So this adhesion to the surface is also very sophisticated. And there is now also different methods that are interesting to be used with bacteria. Besides normal AFM, also like current measurements, magnetic force, electrostatic force. We were talking about also magnetic yesterday and so on. And then of course also as an optical method, the enzyme method. And well, I guess that's what we of course also know, the advantages of SPM in the meantime. Here also what has been discussed yesterday, the combination with optical microscopy that is possible with some of the AFM. And then also nano manipulation. So I mean you had lectures also on cell elasticity, nano indentation, nano manipulation, and various applications, because of course bacteria also have interesting mechanical properties. Sample preparation was also discussed yesterday. There's different methods if bacteria are not adherent. So gelatin, for instance, Cell-Tec, polylysin. It depends on the charge of the bacteria, of course. So mica is, for instance, or glass is kind of negatively charged, but then you can also provide gelatin, Cell-Tec, polylysin. And positively charged surfaces like with polylysin is particularly attractive for negatively charged gram-negative bacteria. So that would be an advantage there. Sample preparation, polylysin is certainly a standard method, and I think one can even buy lysine-coated glass. And then you can use glutaraldehyde to covalently bind bacteria. Yeah, so because this is glutaraldehyde binds to lysines, to both of the polylysine and then also to lysines in the proteins in the bacterial shell. But of course, one always has to be aware, the more chemistry you do, the less chances to destroy the bacteria. And these are also images that can be obtained with AFM, so AFM certainly gives a very high resolution. Another possibility is to filter or trap bacteria, so like to trap them in holes. That is also can be done, which is particularly suitable for round-shaped gram-positive bacteria, for instance. And you can even put them more tightly down by applying mechanical pressure. Yeah, that can be done in holes. Okay. So, and this is the AFM methods. So you can of course use both contact mode imaging or also MEC mode imaging. And that is an example from us from Tanarella. They have this S layer, which I also showed with this walking antibodies that can be recrystallized, so kind of protein-like. And this is what you then finally can achieve if you image in liquids. This was recorded with the tapping mode. Then the resolution can be as high that you can really observe this lattice. There's a S layer, so for surface layer of bacteria, has a periodicity about 10 nanometers in square, but this is a resolution that you can also achieve with MEC mode AFM, for instance. We were also working with curly, and curly are they lead also to cell aggregation and clamping, and they have the ability to adhere to a surface, and this is regulated with adhesive proteins, as you see here. And what they form then, what they develop on the outer surface is this fimbriae. Yeah, so these curly proteins, they can associate to fimbriae, and you can see that here how these fimbriae, they can really be long filamentous structures that can be produced on the outer surface and eases then also the adhesion to surfaces. And you can also manipulate this production. So the protein that is involved in the curly formation is the CSGA. And this can also be genetically induced. So you can induce it. You can induce the expression of the curly, yeah, just genetically. And this is then also how the biofilm formation works. So you can see that here is two types. This is the wild type, and then the CSGA mutant, which overexpresses this curly, and you can follow the formation of the biofilm over time. These are images that are recorded in dry environment in this case, but biofilm formation is something that is really certainly very attractive to follow with the atomic force microscope. Then we also know that we can use antibiotics to destroy bacteria. That was found very early, and the mechanism can also be followed with AFM. So polymyxin is an antibiotics, and if we apply this, that is the structure. If we apply it to the bacterial surfaces, they change the outer membrane because they dissolve into the membrane, and then they finally break the cell, as you see it here. So you can see that the structure of the bacteria is altered before the membrane then finally breaks and the bacteria are destroyed. So this mechanism of the action of antibiotics can nicely be followed with AFM.
So this is like first treated and then embedded on the surface?
No. So basically, we treat here isolated bacteria. Yeah? So basically, you just put a suspension of the antibiotics to the bacteria, and then they bind to the cell membranes, and they penetrate into the bacterial cell membrane and finally destroy them. Okay, so yeah, functional modes for AFM is ion conductance, magnetic force, or electrostatic force. This can be a kind of special AFM methods that are offered also. And then, of course, as also already mentioned, AFM is a very nice force analysis technique, and as such also interesting to be applied not only to cells but also to bacteria. Because from this fimbriae, for instance, what they form is, if we look at it more with polymer science, they form a brush on the membrane of the bacteria, and that can also be very nicely probed. So you can indent into the bacteria, and then with certain models you can analyze elastic and viscoelastic parameters like Young modulus. Yeah, and this is how it looks like. So that is, if we go into if we want to see what these curly proteins do on the bacterial surface, so these are different types of these bacteria. One, the wild type, then one where they lack these CSGA proteins there that produce this curly, and then CSGA mutants before induction and after induction, which the induction is needed that these curlies can produce, and as a comparison on a stiff surface on mica. And we can see that these different mutants behave also differently in their elastic behavior. And we have kind of three regimes that we can tackle. So one is that this polymer brush can be sensed before you actually have contact with the cellular membrane. Yeah? So this polymer brush can be sent. You can see this kind of soft area. And then we have a regime which is here. So here we have some steric force, yeah, that is this region. Basically we have three different regions. One is here, that is the steric region. Then we have here a nonlinear region, and if we indent very deep, we have a linear region. Yeah? So in this linear region gives basically the spring constant of the bacterial cells. And all of these, and the three regions can be modeled. So there's different model, one which models kind of polymer brushes, and then one for the non-linear regime, which gives elasticity like Young's modulus, and then the last one, this linear regime, that can simply be modeled with two springs, basically the spring of the bacterial membrane and the spring of the cantilever. And this is somehow what we get. So, for instance, we can get the Young's modulus. Yeah, that's what we can get from this region here. And you see how that differs. And then the effective spring constants. the effective spring constant is pretty much the same for all, yeah, because you basically you're penetrating already very deeply. But what is different is, for instance, the decay length, which gives you the properties of the polymer brush, yeah, from the curly proteins. And what we see here, we see large differences, wild type and CSGA before induction. But if curly is produced, so like other, then the decay length is feelable. Yeah, so here's clearly brush formation. Surprisingly, for the CSGA, where the CSGA is knocked out, we also feel some polymer brush, but this is not from the curly protein, because if curly is not produced, they form flagella. Yeah? So these are other filaments that bacteria can form. Yeah? So wild type does not form it, so the CSGA knockout mutant forms flagella. The CSGA after induction forms curly proteins, and this can be sensed by the AFM tip before it reaches the actual membrane of the bacteria. And also the Young's modulus is kind of two classes. So there's elasticity, and before induction has pretty much the same Young's modulus, but the knockout mutant and CSGA after induction clearly has then a different Young's modulus. Yeah? So this means that also the expression of the proteins on the cellular membrane kind of changes the elastic properties of the bacteria. Yeah.
Shouldn't be similar. The before induction shouldn't be similar to the minus CSGA.
Here, with the Young's modulus or
The Young's modulus,
yeah,
shouldn't be the before induction, which is 108, shouldn't be similar to CSG minus condition, because before induction means there isn't any CSG on the surface.
theoretically you're right, but this is then again because of the flagella formation, yeah, which we also see. It seems that these flagella are preferably formed from the knockout mutants, yeah, and not if the curly induction is not induced. Yeah?
Okay.
That is the reason. But I mean, from your anticipation, you're certainly not wrong. But this is what happens. That these flagella are only formed from the knockout mutants and not from the other types. Okay. So we were working with many different bacteria, and this is actually the work of Eugene. And one is also Lyme disease. I don't know if there is something like this also exists in India. It does?
I don't know.
Okay. Okay.
In Austria, I think it is there.
Pardon me?
In Austria, you have this.
Yeah, so the region I know where it occurs is Middle Europe, yeah. Also a little bit Scandinavia. And we are working together with Czech Institute from Czech Republic, and also in North America. So these are regions where it comes. So it comes from ticks, yeah. So ticks, they of course they bind to our skin, and what they can do, they can release bacteria or viruses. Yeah, there is the disease with the viruses is called FSME. Yeah, and there is a vaccine against it, against the viruses. But it seems to be more simple to develop vaccines against viruses than to bacteria. Yeah, but they release, besides viruses, they also release bacteria, and so this is just now the symptoms, and yeah, that's where we see where it happens. So it doesn't reach all the way to India, this landscape, but this is where it occurs in Europe. So it's the most prevalent vector-borne disease, in the northern hemisphere. So preferentially in Europe and Northern America. And the bacteria are kind of very interesting because they are spirochates. Yeah? And these spirochates are highly mobile. So, and we can use a positively charged surface and can also image them with the AFM. But they are very fast. Yeah? So, I mean, without fixation, you cannot do anything with the AFM. So if you look at them in the fluorescence microscope, they are highly mobile and they are very fast. And what they do, and this is why they are so dangerous, they are binding to the extracellular matrix. Yeah? And they are also, so this is the host, and they really can stick to there. And from the symptom, it's a little bit similar to malaria, so that the disease can again break out after a certain time. So this means with malaria, it's also the pathogens are still in the body, and can then activate the disease again. And the same is also the dangerous stuff with the Lyme disease. And so this means that it's very interesting to investigate the interaction of Borrelial proteins with proteins of the extracellular matrix. Yeah? So what the binding behavior is and what makes them use the extracellular matrix as a niche for long attachment. Okay? And first of all, what our colleagues did is they did a tick feeding assay. And so this is basically they put the ticks here and fed them to survive them, also with some blood, as you see, to kind of simulate the situation in the body. And then they combined it with a motility assay. Okay. So basically, the bacteria could then penetrate through here. And then they correlated the mobility with the expression of the Borrelial proteins. Yeah? So basically, which proteins make them more mobile in terms of attachment to the surfaces. And this is what they found in this assay. So also to which proteins they are binding. So what you see here is Borrelial proteins, yeah, and then they performed a ELISA type assay. Which of the bacterial proteins bind to which of the ECM proteins? Yeah? You can see the extracellular matrix proteins here: decorin, laminin, fibronectin, and collagen. So these are different adhesion proteins in our extracellular matrix. And then they screened which of the bacterial proteins bind to which proteins. And you can see, for instance, DBPA binds very nicely to decorin, or also DBPB. Okay? And from these screening assays, we then started to do force spectroscopy experiments. Okay? So you saw this. You saw these pictures already, I guess. So we, of course, used our PEG chemistry to functionalize our surfaces with the proteins. And then we performed experiments in which we bound the extracellular matrix proteins, so the different decorin and laminin, where the lysine residue to surfaces. And we used two different Borrelial proteins, DBPA and DBPP, and analyzed them in the methods that I already showed before. And so we found then this behavior, so different conformations. You can also see DBPA and DBPB. We analyzed with laminin and decorin. Okay, so 2 extra ECM proteins and 2 borreliate proteins with all permutations. And we can see here that the strength of binding kinetic off rate is in these ranges, and we can see that particularly DPPA binding to decorin is very long lasting, okay, because the kinetic off rate is just 0.04. With the Bell-Evans model, and but the question is what does this interaction induce between the bacterial proteins and the ECM proteins? Because somehow this is how you could imagine the motility of the bacteria. Yeah? So basically, what you also need to climb up a wall is some holding points. Yeah? And that is how this mobility can be induced, because you need some resistance. If you want to move, then you need some resistance. Yeah? And so what those spirochetes are doing to move along the vessels, that they need some holding points, and this is the binding to the extracellular matrix proteins. Yeah? So they bind there, and then they have some internal motility and can bind faster. Yeah? And this is how this works. So in particular, the proteins that have a very long bond lifetime, they could enhance this movement of the bacteria along the extracellular matrix. Okay, good. What we can also measure is the interaction of bacteria with surfaces. And this is, for instance, in stands, very important, because if you can, I mean, of course, if bacteria grow in your stand, this can cause some severe health hazards. Yeah? So the aim is to create surfaces where bacteria cannot bind, right? So we want to inhibit the infections here, and we tested some different materials, and it's also known that some polymers are synthetic and show a non-toxic, and are also non-toxic, because that's of course also important. And generally, it's of course an important research for biomaterial applications to invent antibacterial surfaces. And this can be, for instance, be accomplished to the polycationic structure of the surfaces, and also polymer brushes. Polymer brushes can also induce an antibacterial effect because they prevent absorption of the bacteria to the surface. So we tested then different coatings. These are polyurethane stands that were functionalized with different polymers. So this, and we tested how these brushes, these different brushes, which have different properties like charge effect and hydrophobic, so they display different charges and also different hydrophobicities, and how these surface coatings are effective against bacterial adhesion. This is how these polymers look like when you produce them. And so what we did is, and this is also something you can do, you can also bind bacteria to AFM tips, and measure interaction of the bacteria with surfaces. And this is what we did here. So we bound E. coli bacteria to AFM tips, and studied their interactions with these different surfaces that you saw in the previous slide. And we measured the binding probability. So we counted how much binding can be accomplished in 100 force distance cycles. And you can see that PU, and so the PU without modification and glass, there the bacteria can bind quite well. So they are not very antibacterial. But then if we use different brushes, you can see the effectiveness, and this surface here is the best, shows the best effectiveness against binding of bacteria. Yeah? So that is, it's kind of a nice method to test antibacterial effectiveness of surface coatings. And then, of course, what you can also do is you can measure the interaction forces. Yeah? So kind of how often they bind and how strong they bind to the surface. And we can also see that this surface that also shows a less binding probability also shows the smallest, if there is an interaction, shows the smallest interaction forces. Okay. Yeah, and this is how these interactions are measured. Okay. Yeah, I think I'm gonna stop here and take, of course, questions from you.
So you pick it up from the surface.
Yeah, we just pick it up from the surface. We functionalize the cantilever with the PEG linkers, acetal linker that bind to the lysins, and then we just pick it up from the surface.
Sir, I wanted to ask that in case of the biofilm formation, why in the initial growing hours, why is the Young's modulus so much higher? Like also
But this was not on the.
this was there.
Okay, but
In the slide where you showed that
Yeah.
curly, like without the CSG operon and the CSG operon being
Yeah.
induced, yes sir,
Uh-huh.
this slide, like here before induction, like why is it so higher?
because there is no curly formed. Yeah, if you don't induce the curly formation, they are lacking the curly structures.
So like they're not forming the biofilm
The
because
formation of the biofilm, that's what we can look here.
if
we compare.
there is no curly, then the biofilm formation will not happen.
Okay, I think I don't have now mutant wild type. I don't have an image now of the biofilm formation after induction, because this is the wild type and this is the knockout. I don't have an image of the curly after induction, and honestly, I mean we did it, but honestly I
These
don't
are
remember.
after induction images.
Yeah, I'm sorry, I don't have. Yeah, sorry, it's here. Yeah.
This is
Ah, yeah,
CSGA plus
this is after induction.
after induction.
Okay, yeah. And the question was, why it doesn't form a biofilm?
Like in the very initial stages of growth,
Yeah.
when there is not much flagellar formation and not much curly formation either, then why is the Young's modulus so high? Like because
Why is the Young's modulus so high?
because here we neither do we have the flagellin proteins nor do we have the curly amyloid secreted. So
No, there is curly proteins. We can also see that here. No, there's curly formation.
no, so before in the very initial hours of growth before induction, I'm saying.
No, this is all after induction. This is just at different times. So this is after induction, yeah, and just monitored at different times. This is just how the biofilm forms over time.
So if you could show the next slide, then I
Okay.
can say.
And there is curly. Okay.
No, probably the previous slide then.
No, I think that. Here
Yes,
the
sir.
data,
Yes, sir. This slide here, I'm talking of the number 108.
Why is it so high?
Yes,
Why-
sir.
why the
The wild type is
yeah
CS.
wild type and CSGA before induction is similar, right? Yeah, and
Why?
and where it's lower is after induction, and this is because of the curly formation.
Yes.
Yeah.
Yes.
And apparently, what happens with the knockout mutant, it of course does not form curly.
But it forms flagellum.
Exactly.
But why is it initially so high? That's what I'm asking.
What you mean with initially?
108 before induction.
Before induction,
Yes.
because it behaves like the wild type. Yeah, it behaves like the wild type. Also for here, you see before induction. I mean, the decay length says something if something is growing on the bacterial surface, right? And this is also very similar for the wild type and before induction. Okay. So this means that no filamentous structures have formed on the bacterial surface. Yeah.
So is it because all the cells are now separate and so they cannot really have that flexible retraction? Is that the reason? Because all the cells are now separate. Neither they have flagellum nor they have curly. Is that the reason?
Right. They neither have flagella nor curly. Right. That's the reason. I don't know why. I also would anticipate that maybe before induction it behaves similar as the knockout mutant, yeah, and as the wild type. I agree. Yeah. I would also rather anticipate that before induction, no, yeah, but it seems to be that, I mean, this is maybe what one would not anticipate, that the CSGA minus forms flagella and the CSGA plus before induction doesn't form flagella. Yeah. This is, I don't know why, yeah, but apparently it seems to be the case.
In case of that COVID virus, the RBDs are acting like calipers, right?
Yeah.
So, sorry. Yeah. So these calipers must be moving in three dimension. It's not just two-dimensional motion.
You mean of the spirochetes? The spirochetes form in three-dimensional.
No. So the image that you have shown in yesterday's lecture.
Okay, you're referring to yesterday's talk.
Yes.
Okay.
So you have shown that in two dimension you showed that the RBDs are moving like this, but these motions would be in z directions as well. When you would have been recording the motion of the RBD.
Yeah, I mean, okay, yeah, sure. I mean, if a spike protein in solution, it would, of course, I mean, there's just that kind of moving like this. But I think what is the important part is how do they move on the surface, because they bind to a cell, and the cell is a surface.
Yeah. So I want to know that if, for an instance, if one of the RBD is in z direction and the cantilever is at that point, will it be able to detect the z motion or the z height from the surface?
You mean now in force spectroscopy or?
No, the tapping mode, high speed AFM only.
Okay.
So, if, for example, if it started moving in the z direction and our cantilever tip is onto the RBD.
If you then you don't see it. Yeah. So I mean, sometimes it depends on the orientation how it absorbs. It also sometimes absorbs sidewise. Yeah. But what we know, for instance, from antibodies, yeah, if we see, or like actually what I showed today, this antibody walking, there we just saw the 2 FAP fragment, but not the FC. Because the FC, if the 2 FAPs are on the surface and the FC shows up, you don't image it because you're kind of floating over it. So you only see, that is with the AFM you only see things that are bound to surfaces. Otherwise it's too flexible and the tip just
Yeah.
doesn't get a signal. Yeah, I mean we do. Yeah, actually you also don't see the in this images. Yeah, we also didn't see the stalk. Yeah, if it's kind of adsorbed downwards. Yeah, but sometimes they are also adsorbing sidewise, and then you see the stalk. Yeah. But somehow things have to be on the surface, otherwise you don't. So yeah, I mean, so it is possible that they also adsorb sidewise, and then you see the stalk, but their preferential orientation is downwards. How they inject it? Now, I mean, what they do, the ticks, yeah, they kind of suck blood out of you. Yeah. So this is what they get from you, and what they give you back to pay for your blood is viruses and bacteria, so that it just penetrates from.
The question is
So in the in
next
vitro, when
slide.
you do it in
couple of slides, I think.
This slide?
For the- yeah,
Yes,
here.
yes.
Here is tick feeding assay.
So
Yeah.
I think how the
this-
collecting the bacteria from the base. How is this done?
The bacteria are here. So basically, this is so you put them here, you feed them, and it is the same. Like let's say this is our skin, yeah. So what they suck the blood, and you can see that here they eat the blood, but at the same time they release bacteria and okay, we don't detect. So these are the bacteria, yeah. So
What is
basically.
that layer? This where they are actually.
This yellowish, this is just the medium, basically. Yeah.
So this one, the SEM image. How did you get the SEM image?
The SEM image, this is something different. This is just to show, this is not from here maybe, this is just, you see this little thing, this is what they punch into you to get the blood, yeah? And this is also what you see here. Yeah, so I guess this is from a skin and a tick. Yeah. But this is this one is this here.